Welcome to the deep dive, everyone. Today we're focusing on something, a pretty dense topic, but super important for you, our medical microbiologist listeners.
Antibiotic action and the big problem these days of resistance.
Exactly. We're going to be kind of like dissecting antibiotics.
Yeah.
Challenges, mechanisms, opportunities.
By Walsh and Wensfitch.
Yeah, that book. It's a tough one, right? So think of this like your shortcut.
So the most important stuff. And maybe some aha moments along the way.
Totally. So even though like you're all experts, I think sometimes going back to the basics can kind of spark new connections.
Right.
So let's start there. Antibiotics. What are they like really at their core?
At their core. Well, they're really just organic molecules, usually pretty small, and their whole job is to mess with bacteria.
Mess with them,
Either killing them outright, that's what we call bactericidal action, or just stopping them from growing, which is bacteriostatic.
And I know as medical microbiologists, you're often like dealing with these serious infections. So
Yeah. You need that knockout punch.
Yeah. So bactericidal is usually the way to go.
Yeah, you could think about it like this. With a severe infection, you're racing against the clock. Bactericidal antibiotics, they get right to the point. They just eliminate the threat. But bacteriostatic, they kind of rely on the host's immune system to finish the job.
Okay, that makes sense. But we can't forget, right, antibiotics aren't working in isolation. They have to deal with this whole, like, crazy complex world of the human microbiome.
Right. And that's a major challenge for sure. We've got trillions of bacteria living in and on us. Most of them, believe it or not, are actually good for us.
Yeah.
But when we use antibiotics, it can disrupt that balance, sometimes in ways we don't expect.
It's like weeding a garden without accidentally, killing the flowers.
Yeah.
Can you give us an example of like how tricky this targeting can get?
Oh, sure. Take Staphylococcus aureus.
Oh yeah, Staph aureus.
Very common on the skin, usually doesn't cause any problems. But it can also be a nasty pathogen, causing anything from minor skin infections to serious bloodstream infections. So the challenge is, how do you hit those bad Staph aureus without wiping out the good ones that keep our skin healthy?
So sometimes those broad-spectrum antibiotics are necessary.
Right.
But they can also be a bit of a blunt instrument.
Yeah, they can. They can wipe out both the good and the bad bacteria, and that leaves the patient vulnerable to other infections, things like Clostridium difficile, which can take over when the normal gut bacteria are gone.
Right. And speaking of troublemakers, that brings us to the infamous escape pathogens.
Oh, yeah, those guys. They're the ones that keep us up at night, the masters of resistance, always coming up with new ways to evade our antibiotics. We're talking Enterococcus faecium, Staphylococcus aureus. Yep, it's a problem child in more ways than one. Klebsiella pneumonia, Acinetobacter baumannii, Pseudomonas aeruginosa, and Enterobacter species.
Wow. That's a whole rogues gallery right there. What makes them so good at evading all of our antibiotic attacks?
Well, they've got a whole bunch of tricks. They can modify the target that the antibiotic is going after, change the lock so the key doesn't fit. They can produce enzymes that just chop up the antibiotic.
Wow.
So it can't do any damage. It's like they have their own little antibiotic degrading factories going on.
It's a constant, like, arms race, isn't it?
Oh, absolutely. It's a constant battle to stay one step ahead. And another one of their tricks, they can actually share those resistance genes with each other.
Share them. How do they do that?
Through something called horizontal gene transfer. It's like they're trading secrets on how to beat our antibiotic.
Okay. So let's go back to basics for a second. The Gram stain. I mean, you medical microbiologists use it all the time. But why is it so important when we're talking about antibiotic action?
Well, the Gram stain, it tells us about the structure of the bacterial cell wall. And that structure has a huge impact on how susceptible the bacteria is to different antibiotics.
Because that outer membrane of gram-negative bacteria, it's like a shield, right?
Exactly. It's an extra layer of protection that many antibiotics just can't get through. That's why gram-negative infections can be so tricky to treat.
So in the lab, how do you actually like measure how well an antibiotic works against a specific bacteria?
One of the key things we do is determine the minimum inhibitory concentration, the MIC.
The MIC, right.
And that's the lowest concentration of an antibiotic that can stop the bacteria from growing.
So a lower MIC means a more powerful antibiotic.
Generally, yes. And we use a couple of different methods to figure that out. One is liquid cultures, where we see if the bacteria can grow in a broth that has different antibiotic concentrations.
Okay.
And the other is agar plate assays, where we actually measure the zone of inhibition around an antibiotic disc.
The bigger the zone.
The more effective the antibiotic.
Got it. Okay, so now let's talk about the antibiotics themselves. We know they come from two main sources, right?
Right. You've got the synthetics, the ones we cook up in the lab, and you've got the naturals, the ones discovered in, well, nature.
And both have played a huge role.
Oh, yeah. Both have been absolutely critical. Synthetic antibiotics, like the sulfonamides, were a game changer back in the 1930s, and we still use them today.
Right. Sulfonamides target folate biosynthesis, which is how bacteria make DNA. It's like cutting off their supply of building materials.
Exactly. Then you've got the fluoroquinolones, another huge success story for the synthetics.
Uh-huh.
They actually inhibit DNA replication so the bacteria can't reproduce. And they're super versatile. They can treat a huge range of infections.
But we can't forget about the naturals. I mean, there was like a golden age of natural antibiotic discovery, right?
Oh, absolutely. The mid-20th century, that era gave us some of the most important classes of antibiotics that we still rely on today. Penicillins, aminoglycosides, tetracyclines. I mean, it's really remarkable how many potent weapons are just hidden away in the microbial world.
It really is. And it shows how, like, discovery and innovation go hand in hand.
Right.
So we're finding these new antibiotics in nature, and then we're using, like, medicinal chemistry to tweak them.
Yeah, make them even better.
Yeah.
Yeah, it's a constant back and forth. We're trying to stay one step ahead of these resistance mechanisms.
And speaking of which, there's this fascinating new concept that's been getting a lot of attention lately, oxidative stress, and how it plays a role in bacterial cell death.
It's a really hot area of research right now.
Okay, break it down for us. What's going on inside those bacterial cells when we talk about oxidative stress?
It all comes down to balance. Cells naturally produce these things called reactive oxygen species, ROS, and they're highly reactive molecules. But cells also have ways to get rid of them, detoxify them. Oxidative stress happens when that balance gets thrown off, when there are too many ROS, and the cell's defenses can't keep up.
So how does that tie into how antibiotics work?
Well, the interesting thing is that a lot of bactericidal antibiotics, even though they might have very different targets, seem to ultimately trigger this oxidative stress response in bacteria.
Oh, wow. So it's like all roads lead to Rome. They all end up at the same breaking point.
Exactly. And this raises some pretty cool questions, like, can we exploit this common pathway to create new antibiotics or even overcome resistance? It's a very active area of research right now.
Yeah, let's get specific. Let's dive into some examples of these natural antibiotic superstars.
Okay, sure.
And how they actually wage war on bacteria.
Sounds good. Let's start with the beta-lactams. Probably the most famous antibiotic family out there.
Oh, yeah. Penicillin. Right.
Exactly. Beta-lactams target bacterial cell wall biosynthesis. Okay. Specifically the formation of peptidoglycan, which is this like mesh-like layer that gives the cell wall its strength.
So they basically weaken the walls, and the bacteria can't hold it together. They just burst open.
Yep, that's pretty much it.
Huh.
And this mechanism is super effective. But the big problem...
I bet I can guess.
Resistance.
Resistance.
The major issue. And it's mostly because of those bacterial enzymes called beta-lactamases.
Those are the ones that break down the antibiotics, right?
Exactly. They break down the beta-lactam antibiotics before they can do their job.
Okay. So what do we do about those bacteria that have figured out how to resist beta-lactams? What are our options?
Well, we do have other classes of antibiotics that can target cell wall synthesis, like the glycopeptide, with vancomycin being the big star.
Vancomycin, yeah.
Yeah.
Often called the antibiotic of last resort.
Right, because it's been amazingly effective against a lot of resistant gram-positive bacteria, including MRSA.
And how does it work?
It basically binds to the building blocks of peptidoglycan and stops them from being incorporated into the cell wall. It's like intercepting the bricks before they can be used to build the wall.
That's a great analogy.
But even vancomycin is not a magic bullet. Bacteria, they're smart. They can evolve.
Of course.
They can modify those peptidoglycan building blocks so vancomycin can't bind to them anymore. Again, it's a constant back and forth.
Okay. We've talked about the ones that go after the cell wall. What about antibiotics that target other essential processes in bacteria, like their inner workings?
Right. Well, another major weak spot for bacteria is protein synthesis. I mean, they're constantly churning out proteins just to survive. And we have a bunch of different antibiotic classes that can mess with that process.
Like what?
Well, there are the aminoglycosides. Streptomycin is a good example.
Okay.
They bind to this part of the bacteria's protein-making machinery called the 30S ribosomal subunit, and they basically cause the bacteria to misread the genetic code.
So they produce faulty proteins, like their instructions get all messed up.
Exactly. It's like introducing typos into their instruction manual.
Hmm.
Then there are the tetracyclines. They also target the 30S subunit, but they work by blocking the binding of something called tRNA, which carries the amino acids to build proteins.
So they're like, what, cutting off the supply line?
Yeah, you could say that. They're cutting off the supply of raw materials that the bacteria need to make proteins. And then there are the macrolides, erythromycin being a well-known example.
Macrolides, yeah.
They bind to the 50S ribosomal subunit. And that prevents the ribosome from moving along the messenger RNA, which basically stops protein synthesis dead in its tracks.
So different targets, same result,
Yeah.
disrupt protein production.
Exactly.
And the bacteria are in trouble. It's amazing how nature has come up with so many different ways to achieve that same goal.
I know. It's pretty incredible, isn't it?
It is. It really is. So before we wrap up this section, I'm just curious, are there any other bacterial processes that are good targets for antibiotics?
Oh, absolutely. DNA replication and RNA transcription are both essential for bacteria to survive.
Okay.
We already mentioned the fluoroquinolones. They target DNA gyrase, which is a key enzyme in DNA replication. And then there's rifamycin.
Okay.
It targets RNA polymerase, which is the enzyme responsible for RNA transcription.
So it blocks the flow of genetic information.
Precisely. It basically paralyzes the bacteria.
Okay, so we've got antibiotics that target cell wall synthesis, protein synthesis, DNA replication, RNA transcription. Am I missing anything?
Well, there's one more important target, folate biosynthesis.
Folate. I thought that was like a vitamin we get from food.
It is, but bacteria can't absorb it directly from their environment. They have to make it themselves.
Oh, okay.
And folate is crucial for making the building blocks of DNA. So sulfonamides, one of the first synthetic antibiotics, they block this pathway. It's like starving the bacteria of what they need to make more DNA.
Wow. I mean, that's a lot of different ways to attack bacteria.
It is. And this is really just scratching the surface. The world of antibiotics is incredibly complex and it's constantly evolving. But, one thing's for sure.
What's that?
Bacteria are incredibly resourceful. They're always finding new ways to resist our best efforts.
Yeah, that's the big challenge, isn't it?
It is. It's a constant battle.
Okay, well, let's switch gears a little bit here and put your knowledge to the test.
Okay.
With some exam-style questions.
Sounds good. Let's do it.
All right, question number 1. Which of the following is a key Y difference between bactericidal and bacteriostatic antibiotics? Is it A, bactericidal antibiotics inhibit bacterial growth, while bacteriostatic antibiotics kill bacteria? Or B, bactericidal antibiotics kill bacteria, while bacteriostatic antibiotics inhibit bacterial growth? Or C, bactericidal antibiotics target gram-positive bacteria, while bacteriostatic antibiotics target gram-negative bacteria? Or D, bactericidal antibiotics are synthetic, while bacteriostatic antibiotics are natural products?
Okay, so the correct answer is B. Bactericidal antibiotics, they actually kill the bacteria, while bacteriostatic antibiotics, they just prevent them from growing. So option A, that's just the reverse of the truth, and C and D, well, those are just wrong. The bactericidal or bacteriostatic action, that doesn't depend on whether you're talking about gram-positive or gram-negative bacteria or whether the antibiotic is natural or synthetic. It's all about how it affects the bacteria themselves.
All right, next question. Which of the following classes of antibiotics targets bacterial cell wall biosynthesis? Is it A, aminoglycosides, or B, macrolides, or C, tetracyclines, or D, beta-lactams?
The correct answer is D, beta-lactams. This class includes the penicillins and cephalosporins, and they work by blocking the formation of peptidoglycan, which is essential for that strong cell wall. Now, the other options, aminoglycosides, macrolides, and tetracyclines, those all target protein synthesis. They work in different ways, but they all mess with the bacteria's ability to make proteins.
Excellent. Moving on to question number 3. Which of the following bacterial pathogens is not part of the escape group? Is it A, Staphylococcus aureus, or B, Streptococcus pneumonia, or C, Klebsiella pneumonia, or D, Pseudomonas aeruginosa?
Okay, the answer is B, Streptococcus pneumonia. Now, the escape pathogens, those are the real bad guys. They're notorious for their multidrug resistance and for causing a lot of those nasty hospital-acquired infections.
They're the ones that give you nightmares.
Pretty much. They include Enterococcus faecium, Staphylococcus aureus, Staph aureus. It makes the list twice because it's just that much of a problem. Klebsiella pneumonia, Acinetobacter baumannii, Pseudomonas aeruginosa, and Enterobacter species. So strep pneumonia, it's a pathogen for sure, but it's not part of that escape group.
Okay, 2 more questions. Here's the next one. What is the primary reason why gram-negative bacteria are generally more resistant to antibiotics than gram-positive bacteria? Is it A, they have a thicker peptidoglycan layer? Or B, they produce more influx pumps? Or C, they have an outer membrane that acts as a permeability barrier? Or D, they mutate more rapidly?
The most accurate answer here is C. They have that outer membrane. It's a tough layer, rich in lipopolysaccharides, and it acts like a barrier, making it really hard for a lot of antibiotics to penetrate and reach their target inside the cell. Now, options B and D, those can definitely contribute to resistance, but that outer membrane, that's the key structural difference between gram-positive and gram-negative bacteria. And just to be clear, option A is wrong. Gram-positive bacteria actually have a thicker peptidoglycan layer than gram-negative bacteria.
Great point. Last question. Recent research suggests that many bactericidal antibiotics induce bacterial cell death by, is it A, inhibiting DNA replication, or B, disrupting protein synthesis, or C, causing oxidative stress, or D, preventing cell wall formation?
The correct answer is C, causing oxidative stress. Now, the other options, those are all legitimate mechanisms for how some antibiotics work, but more and more research is pointing to oxidative stress as a common pathway for a lot of those bactericidal agents. It seems like they're disrupting that delicate balance of oxidation and reduction reactions within the bacteria, leading to a buildup of these harmful reactive oxygen species, and that ultimately leads to cell death.
Okay, that wraps up our exam questions for this section. How did you do out there? Hopefully this was a good refresher, and maybe it even sparked some new insights.
Always good to revisit the basics and make sure we're all on the same page.
Now let's move on to explore in more detail how some of these incredible natural antibiotics actually work.
So you want to dive deeper into these natural antibiotics?
Let's do it.
All right, well, one class that really shows, how clever nature can be is the aminoglycosides.
Aminoglycosides. Streptomycin, that was the first one, right? A real game changer for treating tuberculosis.
Absolutely. Aminoglycosides, they target the 30S ribosomal subunit. That's the bacteria's protein-making machinery.
Right.
And basically, they just jam it up. They cause errors in protein synthesis.
So the bacteria can't produce the protein that they need to survive.
Exactly. And what's really cool is that they can actually get through the bacterial cell membrane, which is a big deal, especially for those gram-negative bacteria, with that tough outer membrane.
Right. But there's a downside, isn't there?
Yeah.
Aminoglycosides, I remember learning about some potential toxicity issues.
That's a really important point. They can have some side effects, especially for the kidneys and hearing. So we have to be careful. Monitoring is key.
That makes sense. Okay, so another class we talked about earlier, the tetracyclines.
Right.
They also mess with protein synthesis, but in a different way.
Yep. Tetracyclines, they also go after the 30S ribosomal subunit.
Okay.
But what they do is block the binding of tRNA. That's what carries the amino acids, the building blocks of proteins, to the ribosome.
Oh, okay.
So it's like they're cutting off the supply line to the protein factory. No more building blocks, no more proteins.
Got it. And tetracyclines, they have a pretty broad spectrum of activity, right? So they're useful against a lot of different infections.
So are. But again, resistance is always a concern. We always got to be thinking about that.
Always. Okay, so how about the macrolides? Erythromycin is one, right? They target the 50S subunit of the ribosome.
That's right. Macrolides, they bind to the 50S subunit, and they basically stop the ribosome from moving along the messenger RNA. So protein synthesis just grinds to a halt.
Oh, wow.
Like putting up a roadblock on the protein production highway.
I like that analogy. So different target, but same outcome. No protein production, and the bacteria are in trouble. It's kind of mind-blowing how nature has come up with so many different ways to do that.
I know, it really is. And it shows just how complex these interactions are between antibiotics and bacteria.
For sure. Okay, so let's switch gears again. What about antibiotics that target DNA replication? Yeah, how the bacteria make copies of their genetic material.
Right. Well, one of the most successful groups of antibiotics that do that is the fluoroquinolones.
Fluoroquinolones?
They inhibit DNA gyrase, which is an enzyme that's essential for DNA replication.
So no DNA replication, no new bacteria.
Exactly. And fluoroquinolones are known for their broad spectrum of activity, and they can get into a lot of different tissues, so they're really useful for a wide range of infections.
But they've also been in the news a lot lately because of concerns about side effects.
Yeah, that's true. We have to be aware of the potential for side effects, tendon problems, sometimes even neurological issues. As with any medication, it's always a balance between the benefits and the risks.
Okay, so we've covered cell wall synthesis, protein synthesis, DNA replication. What about the ones that go after RNA transcription? How that DNA information gets turned into messenger RNA.
Right. Well, one of the real stars in that category is rifamycin.
Rifamycin.
Okay. It's a powerful antibiotic, and it specifically targets RNA polymerase.
RNA polymerase.
That's the enzyme that makes RNA.
Right.
So rifamycin, it basically blocks that communication pathway between the DNA and the protein-making machinery. It cuts off the flow of genetic information.
So the bacteria can't make the proteins it needs.
Exactly. And rifamycin, it's especially effective against mycobacteria.
Including the one that causes tuberculosis, right?
Yep. It's a cornerstone of tuberculosis treatment, a real breakthrough when it was discovered.
Okay, let's shift gears again. I want to talk about that concept we touched on earlier, oxidative stress.
Oh, yeah.
All that buzz in the research world. How does that play into bacterial cell death?
It's really fascinating. We're learning that a lot of these bactericidal antibiotics, even though they have different targets, they seem to kind of converge on this one pathway.
Which is?
Inducing oxidative stress.
Right.
They're all pushing the bacteria towards the same breaking point, even if they take different routes to get there.
I see.
And that's opening up some really interesting questions, like, can we use this knowledge to design new antibiotics?
Or even overcome resistance.
Exactly. It's a really exciting area of research.
And it's really cool that, you medical microbiologists, you're right there on the front lines of this research. You're the ones figuring out how these bacteria work, and you're the ones trying to stay ahead of them.
It's a challenge for sure, but it's also incredibly rewarding.
Okay, you ready for round 2 of exam-style questions?
Hit me with them. I'm ready.
All right, here we go. Which of the following statements about aminoglycosides is false? A. They bind to the 30S ribosomal subunit. B. They inhibit protein synthesis by causing misreading of the genetic code. C. They are effective against both gram-positive and gram-negative bacteria.
Yeah.
They are known for their low toxicity and minimal side effects.
The false one there is D. Aminoglycosides, they are effective against a lot of different bacteria, but they are definitely known for their potential for toxicity, especially for the kidneys and ears.
Oh.
So monitoring is super important.
Okay, good. Next question. Which of the following antibiotics specifically targets RNA polymerase, the enzyme that makes RNA? Is it A, penicillin.B, ciprofloxacin, C, erythromycin, D, rifamycin?
The answer is D, rifamycin. It's a real workhorse. It blocks RNA transcription, so no more genetic information flowing from DNA to RNA.
Okay, next one. Which of the following statements about fluoroquinolones is true? A, they target bacterial cell wall synthesis. B, they inhibit DNA gyrase, an enzyme involved in DNA replication. C, they are primarily effective against gram-positive bacteria. D, they are known for their lack of side effects and excellent safety profile.
The true statement there is B. Fluoroquinolones, they target DNA gyrase, which is crucial for DNA replication. They do have a broad spectrum of activity, and they can get into a lot of different tissues, which makes them really useful. But C and D are incorrect. Fluoroquinolones, they work against both gram-positive and gram-negative bacteria, and while they are generally well-tolerated, they can have side effects, so it's important to be aware of those.
Good point. Okay, almost there. Penultimate question. Which of the following antibiotic classes is often referred to as the antibiotic of last resort, because it's so effective against those resistant gram-positive bacteria, including MRSA? Is it A, beta-lactams, B, aminoglycosides, C, macrolides, D, glycopeptides?
The answer is D, glycopeptides. Vancomycin is the most famous one in that group, and it's been a lifesaver for treating those really serious infections caused by resistant gram-positive bacteria.
Last question. Which of the following bacterial processes is targeted by sulfonamides? Those are one of the earliest synthetic antibiotics, right? Is it A, cell wall synthesis, B, protein synthesis, C, DNA replication. D, folate biosynthesis?
The correct answer is D, folate biosynthesis. Sulfonamides, they block that pathway so the bacteria can't make the folate they need to build DNA.
Okay, that's it for our exam questions for this section. How'd you do?
Hopefully it was a good review and maybe even spark some new thoughts.
Absolutely. It's all about understanding those mechanisms, staying up to date on the latest research. And, even for seasoned medical microbiologists, there's always something new to learn in this field.
It's always changing.
Okay, let's move on to what's arguably the biggest challenge in the world of antibiotics: bacterial resistance. All right, so let's talk about the elephant in the room: antibiotic resistance. It's a global health crisis, and it doesn't seem to be going away anytime soon.
Nope. It's been building for decades, really. A direct result of how we've been using or maybe overusing these amazing drugs.
And we've been talking about how bacteria are like these masters of adaptation, always finding new ways to dodge our antibiotics. But let's break it down. How do they actually develop resistance? What are the specifics?
Well, there are a few key mechanisms, some we've already touched on. One is target modification. Basically, the bacteria change the structure of whatever the antibiotic is targeting, so the drug doesn't work as well, or maybe it doesn't work at all.
So like changing the locks so the key doesn't fit?
Perfect analogy. Imagine a beta-lactam antibiotic. It's trying to bind to a protein in the bacterial cell wall, right? A penicillin-binding protein. But if the bacteria mutates that protein just a little bit, the antibiotic can't bind as well, and the bacteria survives.
Okay, so that's target modification. What else?
Another big one is inactivation. Some bacteria, they produce enzymes that can actually modify or even destroy the antibiotic molecule.
So they've got like a little army of antibiotic destroying ninjas.
I like that. Yeah, they've got their defenses. A classic example is beta-lactamases. Those are enzymes that break down beta-lactam antibiotics like penicillin.
Right, and there are like a ton of different beta-lactamases, right? Some that can like break down a whole range of antibiotics.
Absolutely. That's a big part of why resistance to beta-lactams is such a problem. And it keeps researchers busy trying to develop new drugs that can evade these enzymes.
Okay, so we've got target modification, inactivation. What's the third major resistance mechanism?
Yeah, afflux pumps. Think of them like little bouncers at the bacterial cell membrane. They pump the antibiotics right back out of the cell before they can do any damage.
So even if the antibiotic manages to get in, it can get kicked right back out?
Yep. And some afflux pumps are really good at their job. They can pump out multiple types of antibiotics, so the bacteria become multidrug-resistant.
Wow. So we're stuck in this constant arms race trying to come up with new antibiotics that can outsmart these resistance mechanisms.
It is a race, and one we can't afford to lose. And it's not just about new drugs either. We need to use the ones we have more wisely.
Right. Overuse and misuse. That's a huge part of the problem.
Absolutely. Prescribing antibiotics for viral infections, using them inappropriately in agriculture, people not finishing their full course of treatment, all of that contributes to the problem.
So it's complicated. We need new antibiotics, yes, but we also need better stewardship, education, better global collaboration.
All of that. And that's where you, our medical microbiologist listeners, you're so important. You're the ones on the front lines identifying the pathogens, figuring out the best treatment, educating patients.
Okay, let's wrap up this deep dive with one last round of exam-style questions. Focus on antibiotic resistance this time. Ready?
Let's do it.
Okay, first one. Which of the following bacterial mechanisms is not a major contributor to antibiotic resistance? Is it A, modification of the antibiotic's target; B, production of enzymes that inactivate the antibiotic; C, increased uptake of the antibiotic into the bacterial cell; D, F, the afflux pumps that expel the antibiotic from the bacterial cell?
Okay, the one that's not a resistance mechanism is C, increased uptake of the antibiotic. Bacteria don't want to take in more antibiotics. They want to keep them out. The other three, those are all key ways that bacteria resist our drugs.
All right, next question. The production of beta-lactamases is a major mechanism of resistance to which class of antibiotics? Is it A, aminoglycosides.B, tetracyclines.C, macrolides.D, beta-lactams?
The answer is D, beta-lactams. Those beta-lactamases, they break down those beta-lactam antibiotics like penicillin and cephalosporins. They just render them useless.
Okay, good. Moving on. Which of the following practices contributes to the development and spread of antibiotic resistance?
Yeah.
A, prescribing antibiotics only for bacterial infections; B, completing the full course of antibiotics as prescribed; C, using antibiotics appropriately in agriculture; D, overuse and misuse of antibiotics.
The right answer there is D, overuse and misuse. That's what really drives this whole resistance problem. The other options, A, B, and C, those are all good practices. Those are things we need to be doing to try and slow down the spread of resistance.
All right, two more. Which of the following statements about efflux pumps is true? A, they're protein complexes that help bacteria absorb antibiotics more efficiently. B, they're located in the bacterial cell wall and prevent antibiotics from entering the cell. C, they're highly specific and can only expel one type of antibiotic.
Yeah.
D, they can expel multiple classes of antibiotics, making bacteria multidrug-resistant.
The true statement there is D-efflux pumps can be really versatile. They can pump out all sorts of different antibiotics. That's why they're such a powerful weapon in the bacterial resistance arsenal.
Okay, last question. Horizontal gene transfer. That's the process where bacteria can share genetic material. It plays a huge role in the spread of antibiotic resistance, right? Which of the following statements about horizontal gene transfer is false? A, it allows bacteria to acquire resistance genes from other bacteria, even distantly related species. B, it's a major driver of the rapid spread of antibiotic resistance genes within bacterial populations. C, it occurs only between bacteria of the same species. It can involve the transfer of plasmids, those little circular DNA molecules that carry resistance genes.
The false statement is C, horizontal gene transfer. It's not limited to bacteria of the same species. It can happen between very different bacteria, which is one reason why resistance can spread so quickly.
And that wraps up our exam questions. Hopefully this deep dive has been helpful.
Yeah, maybe a good refresher. Maybe sparked some new ideas.
You know, as medical microbiologists, you all are really key in this fight against antibiotic resistance.
We are. It's up to us to stay informed, to make sure we're using antibiotics responsibly.
Keep learning, stay curious, and keep up the amazing work you're doing.
Absolutely.
Thanks for joining us on the Deep Dive.